Thank you very much. And now for something completely different. Although, in fact, there is a connection between the last talk and this one, and that is smallpox. Because, as you heard, smallpox did destroy the indigenous American society, and also it had a major effect in South America, where it is now thought that millions of indigenous people died from the introduction of smallpox by the Spaniards in the 16th century. So let me talk to you about vaccines in general, and specifically about the future and certain problems. Oh, this photo, my secretary insisted on putting this photo in, and I asked her why, and she said, to prove that you once had hair. So what is a vaccine? A vaccine can be defined as an inactivated, that is killed, or attenuated, that is live but weakened, pathogen, or component of a pathogen, nucleic acid, for example, or protein, that when administered to the host stimulates a protective response of the cells in the immune system. As some of you will know, the cells of the immune system in general come in two varieties called B, Bravo, or T, Tango cells. The B cells produce antibodies in the serum and elsewhere. And the T cells, which originally come from the thymus, produce cells that can kill infected cells. In other words, if someone has an infection and his or her cells are infected, the T cells can come and kill that tissue. Now, this is a quotation from the vaccines textbook, and I think it is very true. The impact of vaccination on the health of the world's peoples is hard to exaggerate. With the exception of safe water, no other modality has had such a major effect on mortality reduction and population growth. Now, when I was young, a long time ago, there were no vaccines given routinely to children. Now, there are at least 16 that I've put on this slide, which you will recognize, no doubt, vaccines such as diphtheria, measles, pneumococcal vaccines. The two at the bottom, Japanese encephalitis and tick-borne encephalitis, are not given to American children because those diseases don't exist here, but they are routinely given to children elsewhere in the world. So now we have a very different situation, but I'd like to go back in history first. And I mentioned smallpox, and how was smallpox conquered? Well, we'll get to Jenner, et cetera, but before that, there was something called variolation. And this basically was taking material from the pus of a smallpox patient and scratching the skin of another individual's with that material and allowing a local lesion to develop in that individual, who would then develop immunity to smallpox. And this was something that was quite important in the 18th century. And I'll come back to that at the end to some degree, but I just want to mention that a very important person in that history was Lady Montagu, who was the wife of the British ambassador to Turkey, and who witnessed variolation being done in Turkey and brought it back to England, where it became well known. The royal family was violated to protect against smallpox. This spread to the United States, as I will come back to later. Voltaire, in his usual mischievous style, in one of his philosophical letters, describes variolation and mentions that it was practiced in the harem of the Sultan to bring women particularly from the Caucasus, where those women were said to be beautiful, and to protect them against smallpox using variolation. So that was a very important beginning to vaccination, because that indeed was what it was. However, there were dangers from variolation. Occasionally, individuals would develop smallpox. And it was Edward Jenner, as I'm sure you all know, who observed that milkmaids were immune to smallpox because they frequently picked up cowpox, obviously from cows. Although now there are some indications that that virus was really what is called today horsepox, but which had spread through the animal population. At any rate, the discovery of Jenner was, as you all know, tremendously important in reducing smallpox incidence. Nothing much happened in the way of vaccines until Louis Pasteur, who was, as you know, a French scientist who worked in a number of fields, not just vaccines. But he became interested in the idea in the 1880s. And what happened was, one summer he left Paris and came back in the fall, and he had left in the laboratory, standing on the counter, a culture of a bacteria that affects chickens, causes a disease in chickens. And that culture, before he left, when injected into chickens, would cause the disease and kill many of them. But when he came back in the fall, he injected a new group of chickens with that material, and nothing happened. Now, most people would have discarded the chickens and so forth, but what he did was he made a new culture of the bacteria and inoculated the same chickens. And those chickens did not develop disease. And the light went on that what had happened was that the bacteria in the culture had been weakened by the heat in the laboratory during the summer. And so he went on to develop a vaccine against that disease of chickens. But of course, he is famous in human medicine for developing rabies and anthrax vaccines. Now, I just want to mention that although the French and the British were certainly the most important vaccine developers in the 19th century, that actually two Americans were also important in the early history, Daniel Salman and Theobald Smith, who developed the first kill, that is, inactivated vaccine against bacteria, using what are today called salmonella after Salman. So then, what is the broad history of the weakened or attenuated vaccines? I'm not going to go through each of these, but just to say that from that original sort of gross physical attenuation using heat or chemicals, it was possible to develop vaccines using passage of the agents in animals or in eggs. For example, the yellow fever vaccine was developed that way. And in more modern times, passage in cell culture allowed for the adaptation of viruses in particular to cell culture, which weakened them by selection, if you will, of the, as you pass the viruses in cell culture, you can select the ones that are less virulent and eventually create a vaccine. On the other hand, as far as inactivated vaccines are concerned, from the original ones, which were, for example, typhoid and cholera, we now have vaccines such as hepatitis A, which is a killed virus. And when it was discovered that certain bacteria produce toxins, the idea of making an antitoxin, that is by inactivating the toxin somewhat, injecting animals or eventually humans with this inactivated toxin, produces antitoxin, which then protects against the disease. In particular, with bacteria, it was realized that they had often polysaccharide capsules, and that if you immunized them with the polysaccharide capsules, you could then protect against that particular bacterium. And one of the members of this society is responsible, Emil Gottschalk is responsible for some of the work relating to a particular polysaccharide vaccine. At any rate, the last point on the bottom right of the slide, gene-produced vaccines, is extremely important because with the advent of genetic engineering starting pretty much in the 1980s and subsequently, it is now possible to use bacteria to produce antigens. And for example, hepatitis B can be produced in yeast or in bacteria. The vaccine antigen itself is produced in vitro and purified and then used as a vaccine. Now, I want to illustrate with 4 vaccines the power of vaccination, starting with, as was said, a vaccine that came from my own lab, rotavirus. Now, rotavirus causes infantile diarrhea, gastroenteritis, and it requires that infants be rehydrated. In this country, something like 50,000 infants a year would have to be hospitalized for rotavirus. And in developing countries, literally hundreds of thousands of children were dying each year because of rotavirus infection. The vaccine was licensed in this country in 2006. And what you see, oops, sorry. Let's see if I can find, yeah, here we go. So it was introduced here, and these are reports of infantile gastroenteritis. These are, in red, are the ones that were tested and proven to be rotavirus, and you can see how they decreased after the introduction of the vaccine. Human papillomavirus, it's sort of a controversial vaccine because you give it to prevent a sexually transmitted disease. How terrible. The viruses, because there are many papillomaviruses, but what they do is they cause a neoplasia. They cause cancer, particularly cancer of the cervix, the uterine cervix, but also other cancers. And the introduction of that vaccine, which was made to protect against the most prevalent types of human papillomavirus, is shown on this slide. And so it protects, incidentally, against genital warts, fine. But more important, it protects against cancer of the vulva, cancer of the vagina. Well, this really should be cervical cancer. And the efficacy is extremely high. It's also important to note that it causes about 50% of cancers of the oropharynx in men, as well as anal cancers. So that now it is recommended for both genders. And it is tremendously successful. And the tragedy is that it is not well accepted. And even in this country, but in some other countries, there have been real problems with introducing the vaccine because of the opposition of certain religions, etc. I also want to mention in passing that there is now a candidate vaccine that actually might treat the cancer once developed, because there are two genes of the papillomavirus called E6, E7, which are responsible for the development of the cancer after the individual has been infected. And what has been done is to make a vaccine of the E6, E7 inactivated, and the pink shows the regression of the cancers compared to the blue, which are the control, the placebo population. And so it appears that one might be able to clear perhaps as much as 50% of the cancers after they've developed. So this is part of the future of vaccination. Meningococci cause meningitis. And as you probably know, that's a terrible disease which can kill within 24 hours or less. In this country, it is now routinely given, and the incidence of meningitis is definitely falling. But I just wanted to show you what's happened in Africa. In Africa, there is something called the meningitis belt, caused by what is called Group A meningococci. And it's a belt that goes right across Central Africa. This was the result of introducing the vaccine against Group A in Burkina Faso. You can see the cases that occurred before the introduction of the vaccine. Then there was a mass vaccination here, and zip. So dramatic effect of vaccination, which is being seen elsewhere as well. Ebola. Well, you all know about the epidemic of Ebola that occurred in West Africa. The pity was, and I'll come back to this, the pity was that although there were vaccines developed in the laboratory against Ebola, they were not in a position to be used. They had not been produced in any quantity. They had not been tested according to the FDA rules, et cetera. And so when the epidemic occurred, there was no vaccine available. A number of companies set to work, and one of them was able to get into Africa with a vaccine. Let me skip that. Get in with a vaccine just before the epidemic stopped. Now, it was considered not ethical to have a placebo-controlled trial in the case of Ebola. But what they did was they introduced the vaccine into one area, and then some weeks later into another area. And what this slide shows is that in the area in which it was introduced first, which is actually the dotted line, within 2 weeks, there was no Ebola. Whereas the area where the vaccine had not been introduced continued to have Ebola, going up, and obviously a strong indication that the vaccine works. I'll come back to infections like Ebola a bit later. I just want to mention in passing that a number of vaccines are now being given in pregnancy. And the idea is both to protect the woman, for example, against influenza, but with a higher efficacy, actually, to protect the unborn infant. Once the infant is born, it has antibodies transmitted from the mother, and that protects the infant for at least several months against neonatal tetanus, for example, against pertussis, whooping cough, and against influenza, because at that early age, the possibility of mortality is quite significant. So, but what about the future and improving? Let me skip a number of slides, which I prepared too many of. But anyway, you all know about influenza and the fact that this year's influenza vaccine is something like 30% effective, which is certainly not enough. And the problem has been that influenza virus changes. It mutates. And it may even mutate between the time people decide to make a vaccine against this particular influenza and when the epidemic actually occurs, because it takes something like 6 months to make the vaccine. So when winter arrives, the individual who is vaccinated is not facing the virus in the vaccine. He's facing a somewhat mutated virus. Well, this is a cartoon of what's called the influenza virus hemagglutinin. Now, the hemagglutinin sits on the surface of the influenza virus, and the vaccines are aimed, the current vaccines are aimed at the head of the influenza hemagglutinin. But the problem is that that's where the changes occur, so, and quite rapidly, actually. So that has been a frustration. But recently, it was discovered that in the stem of the hemagglutinin, there is what's called in parlance an epitope. There is an area there which doesn't change. And the idea now is to use not only the head, but also the stem of the hemagglutinin as a vaccine, which in principle would not require changing from year to year. There, just for information, there are two different stem antigens, but they would cover those two would cover all of the circulating influenza viruses. Another problem that we haven't solved is something called respiratory syncytial virus. This is a virus that is the most important respiratory virus in children, and also next to influenza, the most important in the elderly, where it essentially produces a respiratory disease that can be fatal in someone who's not well otherwise. So it's had #1 respiratory infection of infants, also important in the elderly. Now the problem has been that people have been trying to develop a vaccine against RSV for something like 50 years. And they have failed for reasons which I won't go into. The antigen that has been used is called F for fusion. And it seemed reasonable because that antigen induces neutralizing antibodies. But the vaccine simply didn't work. Well, why? It turns out recent research has shown that the F antigen exists in two forms, which was not realized. One is called post-fusion, meaning that once the virus gets into the cell, that's the form in which it exists, and that's the form that people had been using as the vaccine. Well, on the left of the slide, you see the form of the post-fusion F, which had been used as the vaccine. But in the center, you see what has been recently discovered, that there is a pre-fusion form, which is quite different in structure. And on that pre-fusion form, there are parts, proteins that induce antibodies that are much stronger against the virus than those produced by the post-fusion form. And so this discovery has revolutionized the effort to develop a vaccine against RSV. I also want to mention Cytomegalovirus, the most important virus you've never heard of. And that is, this is something that I've been working on for 50 years with a lot of frustration. But now there's hope, and I'll tell you why. Well, first of all, we showed that a vaccine was potentially possible. That was many years ago. I won't go into that. But why is CMV is important? It's the most common infectious cause of congenital abnormalities. In that sense, it's more important than Zika. It produces microcephaly, deafness, retinitis in the eyes, in the retina, and mental retardation. And so this occurs, the rates are something about between 0.5 and 2% of pregnancies are complicated by a CMV infection because it is a ubiquitous virus occurring both in developed countries and in developing countries. It is also, incidentally, the most common cause of disease in transplantation, both solid organs like kidneys and bone marrow transplantation. So it is a major headache for transplant physicians and patients. So that's the reason for wanting to develop a vaccine. But numerous attempts, including mine, did not succeed until a discovery was made by Tom Schenck, who I believe is also a member of the APS from Princeton. And he, I won't discuss how, but he was able to show that on the surface of the virus, this is a cartoon of cytomegalovirus, there was an unsuspected group of proteins, which are shown by the arrow. I won't bother you with what those proteins do, but one of the things they do is they get together to form a pentamer. In other words, a structure of 5 different proteins. And a group of workers in Pavia in Italy who were studying pregnant women, that is women who are pregnant and are infected by CMV during pregnancy, they showed that a response, an antibody response to this pentamer correlates with protection against transmission. So obviously, this has now become the object of vaccine development, to use this pentamer as part of a CMV vaccine. And I'm optimistic about the future. I want to say something about HIV, which also has been frustrating. There's no natural recovery, strain variation is extreme, and there are other problems that have held up vaccine development. This again is a cartoon of the surface of an HIV virus. And I use it to illustrate the approaches which are now beginning to look possible. On the top, you see something called V3 loop. The V3 loop induces neutralizing antibodies, and so there was a lot of effort into trying to develop antibodies against the V3 loop. And also the CD4 binding site and the membrane proximal region. They were all the object of much work to try to develop a vaccine inducing antibodies against those regions. But none of the trials worked until a trial was done in Thailand. And I must say, it shows you something about science. A lot of people thought that trial shouldn't be done. But it was done, and there was something like 30% efficacy, which is not terrific, but it gave a major clue, and the clue was that antibodies against what's called the V1, V2 region on here correlated with protection, but they weren't the classic neutralizing antibodies. They were antibodies that they're called ADCC antibodies, but I won't bore you with all the details, but they influence what are called natural killer cells and make them work, so to speak. And so now, with that clue, there are trials now going on in Africa with the object of inducing that kind of antibody to see if we can increase the efficacy to at least 50%, if not greater. So, new strategies for vaccine discovery. Again, I don't have time to go through all of these. I just want to mention that one of the things that is of great interest is to use certain viruses like adenoviruses or respiratory viruses, but to insert into those viruses genes from pathogens so that you give the virus to an individual. The virus multiplies to a certain extent, but it also presents the immune system with the proteins from the pathogen. And therefore, the individual responds to those proteins and develops an immune response against the pathogen. This is a type of approach that is now being widely used to develop new vaccines. On the side of inactivated vaccines, I might focus on what's called reverse vaccinology because it's sort of indicates what can be done. Basically, reverse vaccinology boils down to this, that you take a pathogen and you clone it so that you have its nucleic acid. And you break up the nucleic acid into pieces, and you put the pieces into what's called an expression system, which can be bacteria in a Petri dish. The bacteria then produce the product of that piece of DNA, and that expression may result in a protein being produced. And that supernatant from the Petri dish is then inoculated into mice. And one then tests the mice, or the serum from the mice, to see if they have developed an antibody that is protective against the pathogen that you're interested in. And then that could lead, in principle, to a vaccine. So that's one of the techniques, the newer techniques that's being used. Now I want to just mention in passing something called the Coalition for Epidemic Preparedness and Innovation. Because as you know from Ebola and from other infections occurring in developing countries, that major companies are not going to make vaccines against those pathogens. And so in 2015, I and two other people wrote an article in which we proposed the development of a vaccine development fund that would make vaccines that have no commercial interest. And fortunately, this has succeeded in that there is now an organization funded by money from governments and from foundations like the Gates Foundation, which is in a position to make vaccines against this kind of pathogens, that some of which you've heard of, perhaps chikungunya, which is prevalent throughout the world, Lassa fever, which is causing a major epidemic in Nigeria as we speak, and Zika is another one. And so this organization is now working, and we hope will succeed. I will conclude with something called vaccine hesitancy. So-called. For me, it's a euphemism because many of the people who are involved in this are, from my point of view, evil. But anyway, there's growing resistance to vaccination based on ignorance, misconstrual of risks, fear, and lack of altruism. Because the difference between vaccination and other forms of medicine is that not taking a vaccine affects not only you, but your neighbor. Because if you were infected, you can transmit to that person. And so the idea in some communities, usually of well-to-do people, is let others take the risks so my children don't have to. This is a growing problem, and I, among other things, and some of my colleagues are trying to do as much as possible against it. But, you know, it all comes down to the wisdom of the founder of this society, Benjamin Franklin. Of course, at that time, only variolation existed. But Jefferson was variolated himself. George Washington decided to variolate his troops at Valley Forge because the British troops had been variolated, but his troops had not. And Franklin wrote this: in 1736, I lost one of my sons, a fine boy of 4 years old, by the smallpox, taken in the common way. I long regretted bitterly, and still regret, that I had not given it to him by inoculation. This I mention for the sake of parents who omit that operation, on the supposition that they should never forgive themselves if a child died under it. My example showing that the regret may be the same either way, and that therefore the safer should be chosen. And I certainly agree with that piece of wisdom, and I thank you for your attention.
I may want to stay for a couple of questions.
Sure.
Sam Thier from Boston. This audience is aging, and I think it might be of interest to them to know something about the durability of immunity. Pertussis, tetanus, chickenpox, etc.
Well, so that is an important field of vaccinology, so to speak. And I can give no general answer because the vaccines differ. Some of them produce essentially permanent immunity, but some of them don't. For example, a problem that we are facing now is with the pertussis vaccine. There was, many of you will know, a vaccine called whole cell pertussis vaccine, which unfortunately caused a fever and sometimes convulsions. Now, I should mention that convulsions from fever do not leave sequelae. But nevertheless, as you can imagine, parents don't like that, and therefore a new vaccine was developed called acellular pertussis vaccine, which does not do that. However, the new acellular vaccine also doesn't give permanent immunity. The immunity wanes rather rapidly, and so this is a problem we are now currently trying to solve by in a variety of ways, adding what are called adjuvants and so forth. I don't have time to go into all the details, but it is a significant problem. It is why you have to boost certain vaccines. And I would have to go through the list of vaccines and tell you which ones, but there are recommendations made by the Centers for Disease Control for each vaccine, and those recommendations tell you how often you need to have boosters. The one that you will all recognize probably is tetanus, where boosters are recommended every 10 years for people who are at risk.
Thank you.